But welcome everyone to the Special Populations Corps 2023 seminar series of the New Jersey Alliance for Clinical and Translational Science. We really appreciate you joining us for today's presentation. We're very excited to have Dr. Sarah Pachman with us of Princeton University, who's going to discuss the future of families and child well-being study. Before we jump in, if you have any questions during the presentation, please feel free to use the chat for any questions. We'll also have time at the end for Q&A as well. To get us started, I'm going to turn it over to Dr. Dalton Conley, the Henry Putnam University Professor in Sociology at Princeton University and co-lead of the Special Populations Corps of NJACS, to introduce Dr. Pachman. So Dr. Conley, over to you.
Thanks, Kylie. It's my pleasure to introduce Dr. Sarah Pachman, who is currently the Director of Research and Policy at Princeton's Bendheim-Thoman Center for Research on Child and Family Wellbeing. She's NJACS all around because she got her PhD in social work from Rutgers, and her research aims to understand the relationship between childhood contexts, for example, experiences of economic and housing hardship and exposure to proximate neighborhood conditions, and their impact on adolescent well-being, and how policies might serve as levers to mitigate potentially adverse conditions effects. Today, Dr. Pachman will give a brief overview of the core of the future of families and well-being, child well-being study, which some of you may better know by its former title, Fragile Family Study. It's a longitudinal birth cohort study that has followed families for nearly a quarter century, and it's as a oversample of low-income and minority families. She will review available survey and collaborative data sets, including biological indicators and medical records. She'll also provide a summary of analyses that use these data to examine the physical and mental health of mothers and their children using the various components of the Future of Families and Child Wellbeing study. So please join me in welcoming Dr. Pachman. Thank you.
Dalton, thank you so much for that introduction. I'm just going to share my screen. All right. So Dalton, thank you again for the introduction, and thank you all for having me here today. I'm really excited to share with you an overview of the Future of Families and Child Wellbeing study, and particularly some of the health and well-being measures that we have both in our core study and in our collaborative studies. So to give you an overview, I'm going to start by telling you a little bit about what we're referring to as our core study. This core study is what has been going on for the past 22 years. And essentially it was started in the late 1900s, between 1998 and 2000 to be exact, because in the latter half of the 20th century, we saw that there was a significant increase of births in unmarried parents that was occurring in the United States. So over time, we were seeing that more and more children were born to unmarried parents, but we didn't have a data set that would allow us to examine how those kids would fare over time and what their parents' relationships to each other and to the children would look like as the children would grow up. So our original principal investigators, Sarah McClanahan, Irv Garfinkel, and Ron Mincy, decided that we really needed a data set to help us examine those questions. So they started the study with the goal of providing data on 4 key sets of questions. First, what are the conditions and capabilities of new unwed parents, especially fathers? What is the nature of the relationships between unwed parents? What factors push unwed parents together or pull them apart? And how do public policies affect parents' behaviors and living arrangements? And lastly, what are the long-term consequences for parents, children, and society of new welfare regulations, stronger paternity establishment, and stricter child support enforcement? So thus was born the Future of Families and Child Wellbeing Study. As Dalton mentioned, its original name was the Fragile Families and Child Wellbeing Study, and it was named that to acknowledge that while children were being born to unmarried parents, they weren't being born only to single mothers. They were being born into families. We changed the name of the study in the last year to acknowledge that these kids were growing up, and really we were looking at what families would look like in the future. So our name is the Future of Families and Child Wellbeing Study. This study is comprised of a probability sample of about 5,000 births in large US cities. Cities were deemed to be large US cities if they had populations of 200,000 people or more in the mid-1990s. We sampled births in 20 of these large US cities. And then within those cities, we sampled hospitals and then mothers within the hospitals. We have an oversample of non-marital births by 3 to 1. And when using the national weights, the data are representative of births in large US cities occurring from 1998 through 2000. There's a lot more information on our sample. If you're interested, you can check out this sample and design paper. I'm happy to distribute these slides after the presentation. So to give you an idea of what samples are in our sample, or what cities are in our sample, excuse me, here's this map. And so as you can see, we have Newark, one of our cities is in New Jersey. So it's an interesting tie for this group. And we also have cities that are spread out throughout the United States. So this lends itself to researchers conducting all different types of research, looking at differences in state policy or differences in state level characteristics as well. So over time, we have continued to study these children from the time they were born through right now, when we're currently wrapping up our field work at the age 22 survey. So as I mentioned before, we have kind of our core study, and those are the blocks that are in red up at the top. And then we have collaborative studies. And these are studies where other principal investigators have come in and said, hey, I'm really interested in studying this other topic. And our principal investigators have said, okay, that sounds good. Let's piggyback off of this Future of Families and Child Wellbeing study sample. So I'm going to go over briefly what we've collected when, and then I'm going to dive into more detail as the presentation continues. So when our children were born, we interviewed both the mothers and the fathers. And so this is kind of a pivotal moment of the study. Not only was it the study's first wave, Sarah McClanahan had also kind of hypothesized that the moment of birth was this magic moment when fathers had the greatest aspirations of being involved in their kids' lives. And so our interviewers were actually in the hospital when the kids were born, and they'd go in and congratulate the parents on having their baby, and then they would ask them if they could ask them a few questions. And so we started interviewing the mothers and fathers at that time in the hospital room. So it formed a really unique connection with the members of our sample, and we attribute that to being part of the way that we've been able to maintain high response rates over the course of the study. So we continue to interview the mothers and fathers at year 1, so when the child's about 1 year old, age 3, age 5, age 9. And at age 9, we also started to interview the child. Then at age 15, we interviewed the child again, and we also interviewed the child's primary caregiver. So we didn't have enough funding to interview both the mother and the father. So we interviewed the adult who the child primarily lived with. If the child was living with both parents, we interviewed the mother. And then at age 22, we continued to interview the child, and we interviewed that same primary caregiver. Like I mentioned, we're still in our data collection phase for year 22. We're anticipating wrapping that up by the end of this year. Now you'll see in the blue boxes at the bottom of this slide, we have these collaborative studies, and I'm going to be diving into a lot of these in more detail, but you'll see here, this is where we added in medical records, qualitative interviews between a subset of the parents to talk about the parents' relationships to each other, interviews that were both individuals and in pairs. We have in-home observations, we have sleep data, brain data, contextual data, administrative data, and so on. So we have key people who have been involved and in terms of creating the study and then creating these collaborative studies. So as I mentioned, Sarah McClanahan, Irv Garfinkel, and Ron Mincy were the founding principal investigators of the study. Kathy Eden and Jane Waldvogel took over the study a few years ago, and they're now the principal investigators. And we also have a host of collaborative principal investigators who have led all of these other studies. So the sleep study, the cardiovascular study, the medical records, all of that. So they're the ones who are doing these health-related studies are Lauren Hale, Julian Teitler, Orfeo Buxton, Nancy Reichman, Dan Notterman, and Coulter Mitchell. Okay, so you're probably interested, since we've been going on for a couple decades now, in how our response rates are. So over time, we've been able to maintain a pretty high response rate. So these are our mother and father response rates. As I noted on that last slide, we switch over to the primary caregiver interview model at age 15. So I'm going to cover through age 9 right now, and then we'll talk about age 15 after that. So as you can see, among the mothers, about 3/4 of the mothers, regardless of whether they were married to the child's biological father at birth or not, were continued to be interviewed at age 9. Among the fathers, we see that we have higher response rates among the married fathers than unmarried fathers, but still, by age 9, we're reaching about 60% of the unmarried fathers. And again, this is unmarried to the mother at birth. And then 70% of the married fathers were interviewed at year 9 as well. Overall, you can see that we have a fairly high contact rate with the dads over the course of the survey, regardless of whether they were initially married to the mother or not. We're able to talk to 87% of the unmarried fathers and 96% of the married ones. Then we see at year 15, we're able to maintain these high response rates. So we have over 3/4 of the eligible primary caregivers who participated and about 3/4 of the teens who participated in the survey. And we're hoping to continue to have high response rates moving forward in the future as well. Okay, so now I'm going to dive into the data that's available to you. Most of our data is available through public use. However, we do have some additional data that's sensitive, and that's available through restricted use contract data. And those files include things like the residential context, the school context, and biological and health files. So something that's important to note is that all files, with the exception of the genotype array, can be linked to each other using the family identifier. So that means that you can use these biological and health files in conjunction with all of the rich data that have been made available through our core study through those interviews. So as you can see here, most of our data is publicly available. So we have our core surveys with the mothers, the fathers, the primary caregivers, and the children. That's all publicly available. We have in-home assessment data where we actually sent interviewers into this family's homes and did assessments, both of the physical living conditions, of the way that parents interacted with their children, and child assessments as well. We have teacher survey data that's available. We have sleep data, biomarker data, polygenic scores. So all of those are publicly available. We also recently released 2 new public files. So we have our first year 22 data released, actually. So we have COVID data from the SANS subsample. So this is the neurodevelopment collaborative study that Coulter Mitchell leads. And we, in this data, have survey variables from this sub-study. So it's not for our full sample, but I'll go into some detail about the actual sample size in a few moments. And in this data set, we have variables on whether COVID-19 impacted the respondents' mental health and social support systems. And I'll get into more of the measures in that in a few minutes as well. And then we also released some new sleep data. So we already had actigraphy data, but now we also made available daily diaries. And so these diaries are done over the course of approximately 1 week of data collection at year 15. And again, I'll get into that more as well, but this just provides a little bit more color to the sleep data available through the actigraphy data that was already released. Okay, so now for some specific measures. So in our surveys, we collect quite a bit of health data, and this data is collected at every wave. So it's kind of a unique longitudinal source of data from the mothers, the fathers, and the children. We collect data on the respondents' mental and physical health, their disability status, their substance use, any hospitalizations and medical visits, and various health behaviors like fitness, nutrition, and exercise. And then the sleep actigraphy data, as I mentioned before, it's also public use data. It's currently available at years 15 and will in the future, after our year 22 data is ready to be released, be available for year 22 as well. So this is participant level data that corresponds to sleep actigraphy measures across approximately 1 week of data collection at each wave. The data was collected every 30 seconds with a wrist worn accelerometer on the participants' non-dominant wrist day and night for 1 week. And we also, as I mentioned before, have new sleep diary available, which includes sleep, daily activities, and mood. You can see all of our documentation on our website. As I mentioned, I'm happy to make this presentation available to you. And at the end of the presentation, I'll also walk through where you can find all of this on our website. But a little bit more specifically about the variables included in this particular data set, we have things like sleep onset timing at night and during the day. We have naps that were taken, total sleep in a day, waking up after somebody has fallen asleep. So there's a lot of really interesting things here. Also, a public data set is the biomarker data. And the biomarker data is available at year 9 for both the focal child and biological mother, and year 15 for the focal child only. And this was collected through saliva samples. And so at year 9, we did some in-home visits. If there was an in-home visit, then the saliva sample was collected then. Otherwise, it was collected and returned via mail. So these data include telomere length, DNA methylation clocks, and methylation arrays. Next, we have polygenic scores, and we have these scores for a host of things, including body mass index, height, waist circumference, waist-hip ratio, and so on. If you're interested in this, we also have the documentation available on our website. Next, we have that SANS COVID data that I mentioned before. So again, super exciting. It's the first data set that we have out where you can actually use the year 22 data. So this data was collected from participants in the study of adolescent neural development, and it was collected at this very pivotal time during the pandemic. So it's collected from May 2020 through September 2021. And the primary caregivers and young adults received questionnaires that collected information on the impact of the pandemic on social support systems, economic policies, family resources, and mental health. And we have quite a bit of data here. So we have about 560 young adult primary caregiver pairs who were interviewed. And then we also have responses from about 100 primary caregivers and about 140 young adults. So that's like, roughly 800 people who we've been able to make contact with over time, 700. So the data that are available in this data set are anxiety, depression, and perceived stress measures of the respondent support system, and loneliness. And then a few interesting scales or inventories that may have been new to many of us. So the epidemic pandemic impacts inventory, which includes measures of work and employment, education and training, home life, social activities, economic well-being, emotional health and well-being, physical health problems, physical distancing and quarantining, and positive change. And next, there's the Oregon Rapid Assessment of COVID-19 Correlates, which is used to measure several aspects of the COVID-19 pandemic, including pandemic concern, personal health, financial strains, and behavioral changes. The survey also collected data on whether the respondent was an essential worker, what their work was like during the pandemic, so during the time in which they were interviewed, the programs that they had access to, and funding. Lastly, this data set also includes demographic characteristics. Okay. So in terms of the restricted files that are available, so up until now, everything has been publicly available that I've discussed. So it's super easy to get. The restricted data, we have a little bit more of a process, which I'll go into in a few minutes as well. But the restricted use data files that are related to health that we have are medical records. So this is from baseline, candidate genes at year 9, and then genotype arrays at years 9 and 15. Again, the genotype array is the only data set that cannot be linked to other future families' data. So in our medical records, these are the actual hospital medical records from the birth of the focal child. So we had interviewers or researchers who were in the hospitals and were collecting the medical records and actually scanning them in the back of a van. So there's a whole operation where we had people throughout the country trying to gather these data. So the data are available for about 3,700 respondents. You'll notice here that there are about 1,200 respondents that are missing. These missing cases are due to a mix of things. So the hospital not permitting abstracting of records, or there were too cases for it to be financially feasible to do that. The mother refusing consent to provide those records, or that the records couldn't be located in the hospital. These records include several variables, including anthropometric measures of the mother and the baby, the mother's mental health and drug use, and abnormal infant health conditions. Next, we have candidate genes. So these are available at age 9, and they're from saliva samples that were drawn during in-home visits from the focal children and their biological mothers. So we have about 2,900 unique child samples and 2,700 unique mother samples. The variables included in this data set are a series of genotyped candidate polymorphisms. They're all listed out here. And also you can read more about the documentation on the website. Next, for our genotype array, we have genetic data drawn from the year 9 in-home visit and through mail collection at the year 15 study. So these are used with the psych array. And there's going to be data from a multi-ethnic array, which will come out at some point in the next few years. The data include results from genotyping about 3,100 of our focal children. And again, the documentation is available on our website. We have 4 different files that are available. The first file includes the chromosome code, variant ID, position and centum organs, base pair coordinates, the allele codes. And then the second file includes family IDs, sex code, and phenotype value. The third file includes a binary biallelic genotype table, and the fourth file includes the child's sex, parent, and self-reported race and ethnicity, and self-reported or directly measured height, weight, and BMI at age 15. Okay, so I've presented you with a lot of data. As you can see, we have a ton of research that has already been done using the data. And it's really hard to kind of do a comprehensive overview of all of the studies that have been conducted using this data set, because we have these prolific researchers who have published a lot of papers. So what I decided to do for today is just share a few findings with you to give you an example or several examples of how people have used these data and to inspire you to think about your research questions as well. So the first paper that I'm going to tell you about today is called Measuring the Long Arm of Childhood in Real Time, Epigenetic Predictors of BMI and Social Determinants of Health Across Childhood and Adolescence. And this is a paper that the lead author is Laurel Raffington and Dan Notterman and Coulter Mitchell, who are co-PIs on this epigenetic work, are also on this paper as well. So essentially, this paper starts off with the rationale that children who are economically disadvantaged are at increased risk for having a high body mass index and multiple diseases in adulthood. So the authors used longitudinal and twin studies to examine whether epigenetic predictors of BMI developed in adults are valid biomarkers of child BMI and are sensitive to early life social determinants of health. The authors find that salivary epigenetic BMI is associated with children's BMI. And longitudinal analysis suggested that epigenetic BMI is highly stable across adolescence, but remains both a leading and lagging indicator of BMI change. So in some, they find that maternal pre-pregnancy obesity, oh my gosh, I'm so sorry. This should not be on this slide. So I'm sorry about that. So that's the finding there for BMI. I will get to that finding about obesity and asthma in a few moments. Teaser, I'm going to talk about obesity and asthma. Our next paper is looking at sleep, and it's a paper that Lauren Hill and Orfeo Buxton, our collaborative PIs on the sleep study, worked on with Nicole Namode and several other co-authors. And in this paper, looking at neighborhood disadvantage and lower quality sleep and variability in sleep duration. So this study examined the association between neighborhood disadvantage and objective measures of sleep, and finds that adolescents in more disadvantaged neighborhoods spent more time awake after falling asleep, and a greater percentage of nighttime sleep intervals awake, and had less consistent sleep duration than adolescents in more advantaged neighborhoods. Sleep duration and timing did not differ across neighborhood groups, however. So these findings demonstrate that adolescents who live in more disadvantaged neighborhoods have lower quality, less consistent sleep. Next, obesity and asthma. So this is a paper that used data from medical records, and it looked at maternal pre-pregnancy obesity and diagnosis of asthma in offspring at 3 years old. So the objective of the study was to examine pre-pregnancy obesity and asthma. And so essentially what they find, sorry, I'm going to go back to that other slide, is that maternal pre-pregnancy obesity is associated with a diagnosis of asthma in offspring at 3 years old. And the results from the study are preliminary and they needed to be replicated, but they're suggested a complex intergenerational linkage between obesity and asthma. And this relationship was found even when controlling for other demographic characteristics. Okay, so those are kind of some sample findings. And so now I want to go over how you can apply for the restricted use data. So applying for the restricted data is a two-step process, and it includes a preliminary application and a full application. The preliminary application consists of a preliminary application form, a structured abstract, and a CV for each person who will be included in the application. And this will be submitted to ffdata@princeton.edu. Here, I'd like to give a little shout out to our FF Data team who's here with me today. So we have Kristen Katina, who's our Future Families and Child Wellbeing Study Manager. And then we have several of our research specialists, Yu Yang and Pan Yang, along with our postdoc, Michelle Livent. So these are the friendly, smart, and helpful people who will be helping you if you decide that you want to apply for restricted data. The full application instructions are available on our website, and the full application requires an abstract, IRB review, a data protection plan form, signed contract, and then a supplemental research agreement with research staff, and then CV and city certificate for each person who have access to the data. So to give you kind of insight into what's coming down the pike in our world, the year 22 data will be released in the spring of 2024. We also have the Future of Families Generation 3 study, which is being led over at Columbia University. Julian Teitler, Nancy Reichman at Rutgers, and Dan Notterman at Princeton are working on this study. And it includes something that might be of interest to this group: saliva and stool samples from the young adults' children. So the Future of Families Generation 3 is studying the children of our focal children. So this will turn us into a 3-generation study. And we will have the saliva measures from the original parents, the focal children, and then their children as well. So if you'd like to learn more, we have a really robust website where we have all of our public data documentation. So all the links that are in this presentation are to our website. On the public data documentation page, you can learn more about what's in the data. We have the restricted use contract data, where you can learn more about the process of accessing the restricted data. And you can also look at the documentation there. And then we have a couple cool features that I think I'd actually like to show you right now. So we have a metadata explorer where you can check availability across waves, and we have a publication archive where you can see what others have written. So I'm going to share a different screen now and tell you a little bit more about that. Okay. So here we have our website. This is our homepage. And it looks like this is paused. Second. And then, so what we have here is our metadata. And so here I want to show you this because it's really useful for finding different measures over time. So if we look at our advanced search up at the top here, you can look, say you're interested in sleep. If you start searching for sleep, you can see what we have available, and you can also look at measures over time. So what comes up first are our sleep measures from the sleep study, and you can see the data source here, that it's our actigraphy measures. You can see it's at year 15, who the respondent was. And so this is really useful. So you can look and see all of the variables that are available. And then you can see like, okay, well, maybe I want to look at how the parent slept, or how the kid reported sleeping at earlier waves. And all of those are available in here as well. So you can keep on going back in time. So for example, if we want to look at the father's sleep, we can see if the father had trouble falling asleep. We can see the quality of sleep. So again, the part of what makes our study really unique is that we have all of these longitudinal measures. So you can use the metadata to help you find that. And then you can also click on a variable and see if it was asked at other waves. So if there was a similar variable to this, for example, in the past week, how many days did you sleep restlessly? You'd be able to see similar variables asked across different waves as well. Next, so as I mentioned, we have a lot of people who published a lot of articles using our data set. So as you'll see here on our publication archive, which is again accessible through our website right at the top here, you can look up articles that have been published on all different topics. So if you look here, say we want to look at sleep. Before you start working on your project, you can see what other people have already done. And you can see that we have 42 publications that have used our data to look at sleep. So you can scroll through those and see what's already been done. So it's my little website tour for you. And then in closing, I just want to, I'll share my screen again and put up my contact information for you here and the contact information for our data team. So with that, I will open it up to questions and comments.
Thank you so much, Dr. Pachman. I don't see any questions in the chat, but I'd like to start us off with a comment. Almost all my work uses genetic data and looks at the integration of genes and environment. And I know that fragile or future of families, I have to start correcting myself. It had genetic data, but I ignored it for a long time for a couple reasons, but the main reason was because they had genotyped the focal child and genotyped the primary caregiver, I think usually the mom, right?
Yes,
and
correct.
the dads were not genotyped. And in order to have a kind of true natural experiment where the genotype is random on the child, and you want to look at the pure genetic effects and not be confounded with the environment, you need either two siblings so you can difference off each other, and their differences are just the random result of meiosis and segregation of genes, or you need two parents and a child so that you can condition on both parents, and then child genotype is random. And it only had one child and one adult. However, now I'm totally getting into the future of families genetic data because there's a recent development, a methodological tool called Mendelian imputation, where if you know like two siblings, you can impute the average genotype of the parents, or if you know one parent and a child, you can impute the other parents' genotypes due to the nonlinear relationship between the genotypes. It's not just a simple linear combination, and so now you have this great resource in future families where you can sort of get a randomized genotype in the child and see how that, in my case, interacts with the environment, or even conditions their environmental exposures to their own choices and so forth. So I just want to flag that. And it's also most data sets that have genetic data appended to social science studies like health and retirement study, or Ad Health, or Wisconsin longitudinal study, or even like big medical studies like the UK Biobank, don't have enough non-white households, and fragile families future families is one of the data sets that has significant sample size for non-white families. So I think that's really important as we think about sort of the portability and sort of genetic issues that don't translate across populations.
Yeah, that's a great point. And it's great to hear of that development as well, because I think it should be really useful to be able to use our data set with all of the various contextual measures that we have, as well as even some of the more micro level interviewer observations of the actual homes that these kids lived in. Should be pretty useful. So I'm glad to hear that now you're interested in being able to use the data as well.
There's a question for you in the chat too. I don't know if you see it or if you want me.
Yeah. Let's see. So the question is, has there ever been a public education initiative, primary prevention, to educate people about the risks of single parent families along the lines of what has been done with teenage pregnancy and smoking? If so, what were the results? So this is a tricky question. It's kind of outside the purview of this particular study. The only thing that I can think of is kind of some of the marriage initiatives that were brought up during the Bush presidency. So that would be something I'd recommend the questioner to look into. I don't know if anybody else has anything they want to chime in with there.
I mean, I would add that Sarah McClanahan's work, that the original PI and founder of Fragile Families, as it was called, showed a lot that the differences between kids raised in single parent families and dual parent families were mostly a result of the economic differences, not of the marital status per se. Economic and neighborhood and other forms of difference that accompanied different family structures. So that was one of her major contributions. So I think she might be a little, reluctant about policies that only target family structure without targeting sort of the economic disadvantages that go with certain family structures.
Thank you, Dalton. Steve?
Yeah, I just wanted to mention Lena Nepomuski at Rutgers has also been working along the lines of the point that the father's involvement and the effect of policies such as child support enforcement that are arguably counterproductive. So that's an exciting line of work. All this is very exciting. I was wondering about the potential for looking at perhaps gene-environment interactions in development of substance use disorders. And you mentioned the substance use disorder data that you have. Could you talk a little bit about what you have and what's been done?
Yeah, sure. So we do have quite a bit about substance use. So thinking about our children, so we interviewed our kids when they were 15 years old, and now we're interviewing them when they're 22. So kids at age 15 who had already developed a substance use disorder, I think is pretty small portion of the population that we interview. But at age 22, we ask questions, and we also ask these at age 15. So it'll be interesting to have these repeated measures about which types of substances the person uses and how frequently. So I think that we should be able to look at those as outcome variables, using some of the genetic variables as predictors. Does that answer your question?
Did you make any effort to the measures in some of the most widely used data sets like National Survey of Drug Use and Health are actually not so wonderful, but did you make any efforts to classify people as meeting substance use disorder criteria? There's various measures for alcohol, for opioid use disorder, and so forth.
Yeah, we didn't do that. What we did for our measures was we kind of made them parallel to our peer studies, so like Ad Health and NLSY. So they will be comparable across populations that are interviewed by those studies, but we don't do clinical diagnostic criteria here. The only thing that we do that kind of lines up with clinical criteria is the depression inventory that we have. So it's the CIDI inventory.
What is it again? City?
Yeah.
City.
In the city inventory.
So yeah, a PHQ-9 would have been nice, but that probably has a lot of exciting potentials. And of course there you've got, for a life course person, going back to my gerontology life, you've got some really amazing period versus cohort versus environment versus genetic effects going on, because you're measuring this in age 22 interviews took place when in 21.
Throughout the whole pandemic, essentially. So we're still wrapping them up. So we started in 2020, late 2020, and then we're continuing through the end of this year. So we have this interesting rolling period as well for that. So we can look at all different types of exposure and leverage some natural experiments. Some researchers did that with exposure to the Great Recession as well in our
Right.
year 9 wave. So it's a really cool opportunity for researchers too.
Yeah, well, you've got, so many possibilities there, because you've got you've got the exact time, month, and then you've also got, if you want to merge that in, perhaps this is one of the things you're doing in the COVID study. You have sort of where geographically the different waves were hitting, where the school shutdowns were happening, and so forth. So a lot of wonderful geography there with your restricted data.
Yeah, definitely. And we have respondents who live throughout the country now. While everybody was born in 20 cities, now we have respondents who live everywhere in the US. So it's pretty exciting in that way also.
Yeah, the data on, you're familiar, I'm sure, with these, and I should let other people go on, but just as a quick comment, there's these different snapshots that were taken. Like there's an NCHS snapshot that was taken, I guess just a few months or maybe a year into the pandemic, that showed extraordinarily high rates of reporting of distress and depression symptoms. And then it's not clear, that may have leveled off somewhat, but then there's all these data on things like emergency department admissions that soon after the pandemic hit came to represent 7% of all emergency department admissions, suicidal teens, according to one study. So you really have the ability to dig into that and look at the variation over time and space. So that's extremely
Definitely
cool. I look forward to seeing. Is Dan involved in that work, or the?
Yeah, Dan is not involved in the COVID work so much. That's Coulter Mitchell primarily, but Dan is very involved in our study.
Cool.
So looks like we got a couple other questions as well. So the first question from Connor is, when will the census tract data for age 22 be available? So that should be available at some point in 2023. I'm sorry, 2024. And we don't make the actual census tracts available, but we do, we have pseudo tracts, and we include census tract level variables. So stay tuned. You can sign up for the Future of Families newsletter, and then you'll get updates on when data is available. The next question from Mustafa is, how complete is the parental income data in the study? Does it include wages, transfers, etc., and any wealth measures? So I'm going to ask our Future Families team to chime in here if I miss anything as well. But the parental income data, so we have the household income, we have wages, and then we don't have exact measures of transfers, and we don't have wealth measures. We do have questions like, is the household banked? So do they have a bank account? We have questions about credit card debt. We have questions about student loan debt as well, particularly at age 22.
Don't you have measures of whether they own their home or rent?
Yes. Yeah, we have measures if they own or rent their home.
And if they own a car. We have a few other assets questions.
Thanks, Kristen.
And we have we do have some information about receiving like informal transfers of support from family and friends, and receiving support from different public programs like
SNAP and TANF.
SNAP, WIC, TANF, yeah.
And you have home ownership?
Yes, we do.
Yeah, that's important. Wealth would, it's unfortunate that you can't get wealth. Is there any possibility of linking with Social Security earnings records?
We haven't looked into that yet. We've been appending some administrative data. So we have like the National Death Index data that we got. We're in the process of getting National Student Clearinghouse data. But I don't think that we've looked into Social Security. It's something we can definitely look into.
Probably a nightmare, but it might be worth exploring, maybe also healthcare claims as the HRS has done.
Yeah, it'd definitely be interesting. I think the Medicaid data is often at the state level, so it would be tricky, but definitely tricky. Compelling.
And definitely data issues with the federal data. But I was wondering what are there other questions?
I have one really quick question.
Okay.
I really appreciate the sleep data. I think sleep is such an understudied cause and effect of health disparities. Another pet kind of topic of mine that I don't study personally, but I really would love to see more of in these data sets is noise, the noise environment. Are you planning on collecting anything like that?
So that's something, it's funny, Dalton, I recently read an article about this and very much went down that rabbit hole, and such an interesting topic, especially in conjunction with sleep. It's not something that we are pursuing at the moment. Right now, most of our resources are going towards preparing the year 22 data for release, but it's something where, if the funding is there and we have the resources, it's definitely something that's of interest.
Great.
I was wondering if you and Dalton have maybe Dalton, you have some thoughts about again gene environment interactions when you have a shock or a stressor like COVID, and depression. If you've got decent depression measures, so then you've got these various serotonin genes that have been reported to sort of be mediators between those kind of stressors and the outcome of depression. And it sounds like there's a very robust genetic panel. So is that being investigated? Have you thought about that at all?
I am not sure if anybody who's involved in the genetic study is currently looking at that. It's definitely something that would be interesting, and I think one could look at.
It's potentially another really classic test of the the gene environment interaction, where these genes might predispose to depression, but only under particular circumstances of sort of a major shock.
Right.
There's been studies that look at the candidate genes back in the day and interacted it with the Great Recession, the timing, because the timing of which cities got interviewed when is fairly arbitrary and random, and therefore, like when the recession hit was also random with respect to the interviews. So, and they looked at parenting behavior and harsh parenting, but there's a given the staggering of interviews, so that changes the time, makes a time element a bit random. There's no limit to what you can look at with the new genome-wide data and constructing polygenic indices for any range of outcomes. And again, as I added, now that you also have the ability to have a random genetic component in the child, that opens up a lot more possibilities. It's really exciting and endless possibilities.
Oh, that's fantastic. I'd love to hear more as people look into the sort of mental health and substance abuse side, and if there's anything that we could contribute to that, of course, we'd be happy to. We tend to be sort of mindless health services researchers a lot, but this kind of thing is extremely interesting.
I don't see any other questions. Does anybody in the audience have any other questions?
Hi, it's Kristen. I was just going to chime in and say there's definitely opportunities within the COVID sub-study data that Sarah discussed to look at the mental health effects of the COVID pandemic. There's extensive questions on depression and anxiety in that subset of data. So eventually when the well, I guess for the gene environment interactions, the time wouldn't matter. So you could even look at that with the candidate genes data that's already available and look into those interactions for the smaller sample. And then we'll have some more of that information in the full sample once the year 22 data is available. So I'm sure there's folks who are thinking about working on that, but there's definitely going to be a lot of opportunities in that area. And I'll drop the link to the documentation for that COVID subsample into the chat if anyone's interested in more details.
Oh, that's terrific. I'm going to capture that and send it to myself so I can look at it later. Wonderful. So
Great.
Kylie, are we gonna we have 2 minutes, so I guess we should we're Dalton. We should do we have any last questions?
Yeah. Any last question? Time for one, if others. If not, let's all take the time to thank Dr. Pachman for a really stimulating and useful talk. I'm sure you'll be getting a lot of follow-up emails from people with questions, but thank you so much for taking the time to join us today.
Thank you so much for having me, and please feel free to reach out via email.
Thank you so much.
Thank you.